Welcome to Curiosity Corner. Today we're unpacking a groundbreaking study where scientists turned harmless bacteria into cancer-fighting allies. I'll show you exactly how it works. No hype, just facts. Let's start with the big question. Why is this different from existing treatments? Cancer. The word itself carries a weight. A darkness that has haunted humanity for centuries. It's a relentless foe, a thief of health and happiness. A shadow that looms over countless lives. We've poured billions, trillions into research, into treatments. Chemotherapy, radiation, surgery. The tools we have are blunt instruments, powerful but imprecise. They attack not just the cancer, but the body itself, leaving a trail of misery in their wake. Hair loss, fatigue, a weakened immune system. The price of survival is steep, and for many, the treatments fail. Yet even in the face of this formidable enemy, hope persists. Scientists and researchers across the globe toil tirelessly, driven by an unwavering belief that a cure, a breakthrough is within reach. And now, on the horizon, a new weapon emerges. Not from the realm of complex chemicals or radiation beams, but from the microscopic world within us. Bacteria, those tiny organisms we often associate with illness, may hold the key to defeating cancer. This isn't just some wild theory. Researchers at Columbia University, in a groundbreaking study published in Science, have demonstrated the remarkable ability of engineered bacteria to shrink tumors in mice. These aren't your average microbes. They've been genetically modified, transformed into microscopic warriors armed and ready to target cancer cells. The implications of this research are staggering. Imagine a world where cancer treatment no longer means enduring the debilitating side effects of chemotherapy or radiation. Imagine a future where cancer, instead of striking fear, evokes a sense of hope, a belief that it can be defeated with minimal impact on the patient's overall well-being. But how does it work? What makes these engineered bacteria so effective at targeting and destroying cancer cells? The answer lies in the fascinating world of genetics and immunology. Scientists have harnessed the power of CRISPR technology, a revolutionary gene editing tool, to modify harmless bacteria, turning them into cancer-killing machines. The results, at least in mice, have been nothing short of astounding. Tumors have shrunk dramatically with minimal side effects. The engineered bacteria, armed with specific proteins that mark cancer cells for destruction, act like guided missiles, leaving healthy tissue untouched. This is a paradigm shift in cancer treatment, a move away from the blunt force of traditional therapies to a more precise, targeted approach. But as with any groundbreaking discovery, questions arise. How safe is this technology? What are the long-term effects? Can these results be replicated in humans? The journey from promising research to a widely available treatment is long and arduous. Rigorous clinical trials are needed to ensure both efficacy and safety. Ethical considerations must be addressed, and the potential impact on healthcare systems worldwide carefully evaluated. The fight against cancer is a battle waged on multiple fronts, with an arsenal of treatments constantly evolving, in response to the disease's relentless adaptability. Yet, despite decades of research, and billions poured into developing new therapies, the tools at our disposal remain imperfect, each carrying its own set of limitations and unintended consequences. Chemotherapy, the mainstay of cancer treatment for decades, relies on powerful drugs to kill rapidly dividing cells. While effective in eradicating some cancers, it often fails to discriminate, attacking healthy cells alongside malignant ones. Radiation therapy, another cornerstone of cancer treatment, uses high-energy beams to destroy cancer cells. But it too can damage surrounding healthy tissue, leading to a host of debilitating side effects. These traditional treatments, while sometimes life-saving, often come at a high cost, leaving patients weak, vulnerable, and grappling with long-term health issues. The quest for more precise, less toxic treatments has driven researchers to explore new frontiers, including immunotherapy, which harnesses the power of the body's own immune system to fight cancer. Chemotherapy, a double-edged sword. Chemotherapy's effectiveness lies in its ability to target rapidly dividing cells, a hallmark of many cancers. However, this indiscriminate attack on dividing cells also affects healthy tissues that rely on rapid cell division, such as hair follicles, bone marrow, and the lining of the digestive tract. This collateral damage manifests in a range of debilitating side effects, from hair loss and nausea to a weakened immune system, leaving patients susceptible to infections. Furthermore, chemotherapy's efficacy can wane over time as cancer cells develop resistance to the drugs, much like bacteria evolve resistance to antibiotics. This acquired resistance poses a significant challenge, forcing oncologists to constantly adjust treatment regimens, often with diminishing returns. The search for new chemotherapy drugs and strategies to overcome resistance is an ongoing battle, one that highlights the limitations of this approach. The development of targeted chemotherapy drugs, designed to specifically target cancer cells while sparing healthy ones, has offered a glimmer of hope. These targeted therapies, however, are not without their own challenges, including drug resistance and a limited range of cancers they can effectively treat. Radiation. Collateral damage. Radiation therapy uses high-energy beams to kill cancer cells, but can damage surrounding healthy tissues. Side effects include skin irritation, fatigue, and increased risk of secondary cancers. Advances like intensity-modulated radiation therapy improve precision, but risks remain. Balancing effective treatment and minimizing damage is crucial. Immunotherapy's reach and grasp. Immunotherapy harnesses the body's immune system to fight cancer, offering hope for long-term remission. However, it's not effective for all cancers or patients, and can cause severe side effects. Ongoing research aims to expand its reach and improve outcomes. Cancer's ability to evolve, adapt, and evade the body's defenses makes it a formidable foe. Traditional treatments often fall short due to toxicity, lack of precision, and drug resistance. The search for new, effective cancer treatments is driven by the need for precision therapies. Cancer's heterogeneity and constant evolution make it a moving target. Developing one-size-fits-all therapies is challenging due to cancer's complexity. The search for new therapeutic targets is crucial for effective treatments. The human body is a universe unto itself, teeming with trillions of bacteria that outnumber our own cells. While some bacteria are associated with illness, many are beneficial, playing crucial roles in digestion, immunity, and even mental health. These microscopic organisms, long viewed with suspicion, are now being recognized for their therapeutic potential, particularly in the fight against cancer. The concept of using bacteria to combat cancer is not entirely new. In the late 19th century, William B. Coley, a surgeon at Memorial Sloan Kettering Cancer Center, observed that some patients with inoperable tumors experienced remarkable regressions after developing post-surgical infections. He hypothesized that the infections stimulated the immune system to attack the tumors, a phenomenon now known as the Coley's toxins effect. While Coley's methods were crude and often dangerous, his observations hinted at the potential of harnessing the immune system to fight cancer. Today, armed with a deeper understanding of immunology and the tools of genetic engineering, scientists are developing sophisticated approaches to transforming bacteria into targeted cancer therapies. These engineered bacteria, acting like microscopic Trojan horses, can infiltrate tumors, deliver therapeutic payloads, and rally the immune system to join the attack. CRISPR technology allows scientists to edit DNA with precision, transforming bacteria into cancer-fighting agents. CRISPR, a natural bacterial defense, is now a powerful gene editing tool. Scientists use CRISPR to engineer bacteria to display CD47, a protein on cancer cells. These engineered bacteria act as decoys, helping the immune system attack cancer cells. The cancer battlefield. Seeking hypoxia's embrace. The tumor microenvironment is a hostile landscape, where cancer cells rapidly outstrip oxygen and nutrients, creating hypoxia. Hypoxia drives cancer cells to adapt, increasing angiogenesis and shifting to anaerobic metabolism. Engineered bacteria can target hypoxic tumor regions, delivering therapeutic payloads and enhancing treatment efficacy. Unleashing the immune system. A two-pronged attack, the human immune system holds immense potential for fighting cancer. However, cancer cells often evade or suppress the immune system. Engineered bacteria can modulate the immune response to target cancer cells. One approach involves engineering bacteria to produce cytokines. Cytokines stimulate immune cells to recognize and destroy cancer cells. This creates a localized inflammatory response, boosting anti-tumor activity. Another strategy involves engineering bacteria to express tumor-associated antigens. This primes the immune response to target cancer cells throughout the body. Cancer immunotherapy has shown promise, leading to long-term remissions. From mice to mankind. A glimmer of hope. Engineered bacteria show promise in cancer therapy, with mice studies demonstrating tumor regression and improved survival. These bacteria target tumors, deliver therapies, and stimulate the immune system. Translating these results to humans is complex, due to immune system differences. Despite challenges, the potential benefits are significant. Clinical trials are underway to evaluate safety and efficacy in humans. Engineered bacteria for cancer therapy represent a paradigm shift, offering precise, personalized treatments. However, potential risks and ethical considerations must be evaluated. Scientists are developing kill switches to control or eliminate bacteria if necessary. Rigorous safety testing and long-term monitoring are crucial. Ethical principles must guide development, prioritizing patient safety and informed consent. Engineered bacteria hold the promise of transforming cancer treatment, offering new hope to millions of patients worldwide. The idea of introducing bacteria into the body as a treatment, even engineered ones, might understandably raise concerns. However, the researchers at Columbia University have taken a safety-first approach in designing these cancer-fighting microbes. One crucial safety feature is the incorporation of kill switches into the bacteria's genetic code. These kill switches are essentially self-destruct mechanisms that can be activated remotely, ensuring they don't linger beyond their intended purpose. Furthermore, these engineered bacteria are designed with a limited lifespan, programmed to self-destruct after a certain amount of time, ensuring the bacteria's therapeutic effects are transient and controlled. While the preclinical data in mice paints a promising picture of safety and efficacy, the leap from animal models to human trials is significant and fraught with potential unknowns. The human immune system, far more complex and nuanced than that of a mouse, could react to these engineered bacteria in unexpected ways. Allergic reactions, while not observed in mice, are a possibility, as are unforeseen immune responses that could lead to inflammation or other adverse effects. Another concern is the potential for these engineered bacteria to trigger autoimmune responses. Addressing these unknowns will require extensive long-term monitoring of patients treated with this therapy, carefully tracking any potential side effects or unexpected outcomes. The journey from a groundbreaking scientific discovery to a widely available medical treatment is often long and arduous, marked by rigorous testing, regulatory hurdles, and the need for robust data to demonstrate both safety and efficacy. The engineered bacteria developed at Columbia University, while incredibly promising, are still in the early stages of this journey, with human trials slated to begin in 2025. These initial phase 1 trials will focus primarily on safety, evaluating the therapy in a small group of patients with advanced cancers who have exhausted other treatment options. If these initial trials prove successful, larger phase 2 and phase 3 trials will follow, evaluating the therapy's efficacy in treating specific types of cancer and comparing its effectiveness to existing treatments. Assuming the engineered bacteria continue to demonstrate safety and efficacy in clinical trials, it's estimated that FDA approval could take anywhere from 5 to 10 years. The potential for engineered bacteria to revolutionize cancer treatment extends beyond their effectiveness in targeting and destroying cancer cells. Unlike costly and complex treatments like CAR T-cell therapy, bacterial-based therapies offer the potential for a more scalable and affordable approach to cancer treatment. Growing bacteria is, in essence, a fermentation process, similar to brewing beer or culturing yogurt. Experts estimate that bacterial-based cancer therapies, if proven successful, could cost up to 80% less than CAR T-cell therapy, which currently runs upwards of $500,000 per patient. This dramatic cost reduction has the potential to make this groundbreaking therapy accessible to a far wider range of patients, particularly those in low and middle-income countries where access to advanced cancer treatments is often limited. The development of engineered bacteria as a weapon against cancer is undeniably exciting, offering a glimmer of hope in a field often marked by difficult choices and uncertain outcomes. The preclinical data is compelling. The science is sound, and the potential benefits, both in terms of efficacy and affordability, are immense. However, it's crucial to temper this excitement with a healthy dose of caution and a commitment to rigorous scientific scrutiny. The journey from promising research to a widely available treatment is paved with both triumphs and setbacks. The potential of engineered bacteria to transform cancer treatment is undeniable, but translating its promise into tangible benefits for patients will require patience, perseverance, and a steadfast commitment to scientific rigor. The use of genetically modified organisms in agriculture has been a subject of intense debate for decades, often sparking controversy and fueling public mistrust. While genetically modified crops have become commonplace in our food supply, the idea of introducing engineered bacteria into the human body as a medical treatment is a concept that raises a new set of ethical concerns and societal anxieties. The engineered bacteria developed for cancer therapy have been designed with multiple layers of safety measures, including kill switches and limited lifespans, to mitigate the risk of unintended environmental or health impacts. Bridging the gap between public perception and scientific reality will be crucial in gaining widespread acceptance for this promising new technology. Cancer is a global health crisis, affecting millions of people worldwide, regardless of race, ethnicity, or socioeconomic status. However, access to advanced cancer treatments, such as CAR T-cell therapy, is often limited to patients in high-income countries, leaving millions in low- and middle-income countries with few treatment options and a bleak prognosis. The potential for engineered bacteria to provide a more affordable and accessible alternative to costly cancer treatments has profound implications for global health equity. Unlike CAR T-cell therapy, which requires specialized facilities and highly trained personnel, bacterial-based therapies can be produced at scale using relatively simple and inexpensive fermentation processes. The development and deployment of engineered bacteria for cancer therapy represent an opportunity to bridge the global cancer care gap, providing hope and healing to millions who currently lack access to life-saving treatments. The advent of engineered bacteria as a medical treatment marks a pivotal moment in the history of medicine, ushering in a new era of possibilities and challenges. As we stand on the cusp of this new frontier, it is imperative that we proceed with both enthusiasm and caution, embracing the potential of this technology while carefully considering its ethical implications and societal impact. The ability to engineer living organisms for therapeutic purposes raises profound questions about the nature of life, the limits of human intervention, and the potential consequences of manipulating the building blocks of biology. While the potential benefits of this technology are immense, it is crucial that we engage in open and transparent dialogue about the ethical considerations, ensuring that the development and deployment of engineered bacteria for cancer therapy are guided by principles of safety, efficacy, and social responsibility. The future of medicine is being shaped by groundbreaking innovations like engineered bacteria for cancer therapy, and it is our collective responsibility to ensure that these advancements benefit all of humanity. The war against cancer has raged for centuries, a relentless battle against an insidious foe, and now, from the unassuming world of bacteria, a glimmer of hope emerges. A potential weapon unlike any other. The idea of harnessing the power of bacteria, those microscopic organisms often associated with illness, to fight cancer might seem counterintuitive, even paradoxical. Yet, science has a way of revealing the extraordinary within the ordinary, of transforming our understanding of the natural world and unlocking its hidden potential. Imagine a world where cancer treatment no longer means enduring the debilitating side effects of chemotherapy or radiation, where the immune system, empowered by engineered bacteria, becomes a formidable force against malignancy. The need for vigilance and vision. The road ahead is long, paved with promise, and potential pitfalls from research to treatment. Challenges abound and requiring rigorous testing. And patient safety. Vigilance is key. As we introduce engineered bacteria into the human body, careful monitoring for side effects and long-term consequences is paramount. Transparency and adaptability ensure safe development. Vision and transformative potential drive this journey. Embrace the future of personalized cancer treatment. Embracing the future, we're one microbe at a time. Engineered bacteria for cancer therapy is a testament to human ingenuity and determination to conquer disease. Solutions to medical challenges often lie in unexpected places, discovered by the curious and courageous. As we embrace this new frontier, let us balance optimism with caution. Thank you for joining me on this exciting exploration. Remember to like, comment, and subscribe for more fascinating dives into the world of science.